Hi, welcome back to Human Metabolism, Chapter 3, Part 2 for Monday, June 8th. Last time we talked about chemical reactions that take place in the body in order to keep us alive, things like hydrolysis, phosphorylation, and redox reactions. We also described cellular work. There are 3 different types, and how enzymes, which are specialized catalytic proteins, help push a reaction in the forward direction faster in order to produce products from a set of reactants. We should also have an appreciation for the universal energy source we introduced last time, called ATP. The key here is, how do we make lots of ATP energy? If it's universal, then it must be used a lot. Our brain will need ATP even when we're sleeping. Our GI system will need to digest our food, and certainly our muscles will need lots of ATP energy during locomotion or heavy work. Human metabolism starts with the consumption of food, and foods that contain glucose, one of our simple sugars from Chapter 2, is a preferred energy source for nutritional energy. Let's first describe how we burn glucose in a process called glycolysis. The burning of glucose, or glucose oxidation, requires oxygen, but the first step in this long process is able to run with or without oxygen, which is great if you're running a marathon or doing other heavy work. Glycolysis can run in the presence of low oxygen by shifting to a process called fermentation. I'll show you that in a process in a little bit later in these notes, after we cover glycolysis first. Glycolysis can be seen as the general chemical equation here. Glucose, our 6-carbon sugar, combined with 6 oxygens, will produce CO2, water, and ATP energy in a balanced chemical equation. So here we can see the stepwise cycle of glucose breakdown, which is the first step in gaining universal energy in the form of ATP. Heat is released along the way with energy needed for the next step in the process, which is a phosphorylation reaction that places a terminal phosphate group on the end of an ADP to give us ATP. So adenine diphosphate becomes adenine triphosphate, ATP, our universal energy source. You can refer to figure 313 in your text. Glycolysis is a 10-step process controlled at every step of the way by an enzyme that will only participate in a single reaction. Glycolysis can only be run in the cytoplasm of cells, and the products of glycolysis will enter into the mitochondria for the next 2 important steps in this process of complete oxidation of glucose. The overall reaction for this process adds NAD, ADP, and inorganic phosphates to the left side of the equation, which means these are reactants, not products. On the product side of this equation, we see that 2 ATP are produced from this 10-step enzymatically controlled process. On the product side, we have 2 pyruvates and the reduced form of NAD, called NADH, takes on a hydrogen. We also have some water and 2 protons on the product side of glycolysis. So in this case of glycolysis, you can think of the old saying that it takes money to make money. And that's the case here. So glycolysis produces 4 ATP, but 2 of them are needed to run the first 2 steps that kick off this entire process. So we only gain 2 ATP. That's your net pay. In figure 315, this shows the 10-step process in a simplified form of glycolysis, and in summary, we can see that glycolysis can be linked to the Krebs cycle through the 2 pyruvates that were produced along with our ATP. The next 2 major steps after glycolysis take place in the mitochondria. Remember, that's the organelle known as the powerhouse of the cell due to its massive amounts of ATP production when oxygen is available. Pyruvate is a 3-carbon product of glycolysis, which splits a 6-carbon glucose into 2 3-carbon pieces. As pyruvate moves into the mitochondria, it gets modified further into a 2-carbon molecule called acetyl coenzyme A, or acetyl-CoA. You may also see it written as A-CoA. This 2-carbon acetyl-CoA is what actually enters the Krebs cycle. In figure 315, 316, and 317, use your text to see these illustrations on how these two important reactions are linked together. So we've covered glycolysis, and the pyruvate product of glycolysis is now being used to feed directly into the Krebs cycle, which is step 2. Our 3-carbon pyruvate is modified by losing a carbon in the form of CO2. What do we do with excess CO2 in the body? Okay, so we're reducing the carbon skeleton, highlighted here in the pink box, from a 3-carbon pyruvate to a 2-carbon acetyl-CoA. And in figure 317, we see the acetyl-CoA entering the Krebs cycle here at the top, and it meets with oxaloacetic acid from the citrate to form citrate. Oftentimes, Krebs cycle is called the citric acid cycle. So these products in this cyclic process are used to regenerate themselves. So that's why Krebs cycle is usually drawn as a circle, because it's continuous. Krebs generates 2 molecules of CO2, and those are going to be exhaled off. They come off of this cycle, and with each turn, this modifies the carbon structures of the other molecules involved in this process. Krebs cycle is not a huge money-making process, so we're not gaining a lot of ATP here. Depending on what research you read, there's 0 to 1 ATPs made here. Krebs is a prep phase for the big payoff, which occurs in our last step, step 3, aerobic respiration. The Krebs cycle supplies 8 of the 12 required coenzymes needed for the final step in this 3-step cellular process called oxidative phosphorylation, which we'll talk about next. This is an oxygen-requiring step that occurs in the mitochondrial matrix and generates 34 ATP per glucose. NADH and FADH2 will act in the redox reactions in step number 3. This would be more of the complete burning of glucose. Remember, we've only made 2 or 3 ATPs so far with glycolysis and Krebs cycle. So now let's talk about the payoff phase. In order to make lots of ATP, we need to understand the electron transport chain, or ETC. Figures 319 and 320 illustrate this process. In aerobic respiration, this is carried out by a series of proteins highlighted in this teal blue box. They're embedded in the mitochondrial matrix. These proteins are responsible for the electron transport process. They form a chain reaction by donating their electrons to the next protein in line. Remember Leo, the lion, goes grr? I gave you that acronym in a previous lecture set. Well, if an electron is lost, then the protein becomes, so loss of an electron, Leo, that's oxidized. If an electron then is donated or lost, then the neighboring protein gains the electron, therefore grr. Gain of electron reduced. This goes on all day long, basically until you die. This is where 34 ATP are generated, because at the end of the line there is an enzymatic protein called the ATP synthase. The suffix ase almost always indicates this protein is acting as an enzyme. When we look inside of the matrix in figure 320, we see the process in action. The outside video links that I've included on this page will help you a lot in understanding this very important process, because other videos will put these molecules into motion, and this may help you understand this final process called chemiosmotic coupling. This is a theory that won the Nobel Prize in the 70s, and it has launched human physiology really into the next level. This theory describes how electron transport from protein to protein to protein in a chain or series is linked to the massive ATP production process. The secret is in the accumulation of these hydrogen ions, these protons that are built up. They're indicated in this picture as little blue circles, okay, outside of the inner membrane. So what happens is these protons build up in the intermembrane space. This accumulation of protons from the electron transfer process creates a concentration gradient. We will use this concept more in next week's lab number 2. A high concentration of any molecule will naturally want to go to a state of lower potential energy, just like rolling a ball down a hill. The flow of protons through the ATP synthase drive the production of massive amounts of ATP, like a motor. Oxygen is going to be the terminal electron acceptor. So without oxygen, this process does not work. Oxygen will get reduced to become water. When oxygen is not present due to a deficient amount of oxygen, usually during heavy work in the body, lactic acid tends to accumulate. So what happens then is we get into an anaerobic process, and lactic acid will build up in the muscles and the electron transport chain shuts down. Our terminal electron acceptor is no longer available. So we have to go back to our only option, and that is glycolysis that we studied in step 1. Glycolysis is able to run without oxygen, as shown here in figure 322. Here we can use the reduced form of NAD to become oxidized, so pyruvate becomes lactic acid. We still produce 2 ATPs using this process. This reaction is why you feel tired after heavy exercise, muscles feel sore the next day, and you remain out of breath for a while as you recover. Removal of lactic acid from the muscles happens through a process called the Cori cycle, shown here. I believe the 6th edition of your textbook has eliminated this diagram, but luckily I still have it here to share with you. The blood flow away from the muscles pulls the lactic acid out towards the liver. Muscles are indicated in the circle on the left, and the liver is on the right. So the blood flow pulls these toxins, really. Lactic acid is what causes muscle pain. Pulls them away from the muscle towards the liver. So we're using the bloodstream. And then when it hits the liver is really well equipped to handle the conversion of lactic acid into a stored form of energy called glycogen. This polymer was introduced back in chapter 2. On the right side of this diagram, the liver uses its enzymes to convert lactic acid back into pyruvate, where it can be phosphorylated, and the 2 pyruvates into glucose through the 6-carbon phosphorylation. So G6P is often abbreviated for glucose 6-phosphate. You see that in both muscles and liver. So the G6P, or glucose that's been phosphorylated, can either be immediately used by leaking glucose back into the bloodstream, or it can go up another level, right up to the top, below the word liver. It can be stored as glycogen. So depending on the body's demands, you have this recyclable form of energy through lactic acid. This is usually the point where a cool down, easy bike for 10 minutes or a walk is really helpful to prevent muscle soreness the next day, because you're able to flush that lactic acid out of the muscle, get it into the bloodstream here in step 4, and take it directly to the liver. The body has multiple ways of extracting energy from the foods you eat. So when sugar is not around, the body will shift towards burning fats or proteins. Notice how you still have the central reaction of the 3 steps we just discussed. At the top, I've circled in red the 3 substrate energy sources from foods. Glucose is still central to human metabolism, but the other 2 optional reactions will feed into Krebs or oxidative phosphorylation by way of pyruvate or acetyl-CoA. So proteins, as a fuel source, here on the left, are modified by first cleaving off the nitrogen group, which is called a deamination process. These amines are toxic to the body and will get eliminated as urea and taken care of by the kidneys. After this modification, the amino acids can feed directly into Krebs as pyruvate, acetyl-CoA, or oxaloacetic acid in the Krebs cycle. Fats, over here on the far right, are modified through a process called beta oxidation. This yields two carbon chunks, two carbon pieces off these long chain fatty acids or triglycerides. You get these two carbon pieces that are broken down and can feed directly into the Krebs cycle, mainly by way of acetyl-CoA, which is a two carbon molecule. The skeletal muscles also contribute as alternate fuel sources because they store glycogen just like the liver does. So in times of demand, they have their own energy source. Glycogen is broken down into glucose in a process called glycogenolysis. This frees up glucose and can start the ATP making process at the 10-step glycolysis step, step number 1. There's also a process of making new sugars from non-carbohydrate resources, proteins and fats, and this is called gluconeogenesis, so neo meaning new. And the liver is great at doing this. Fats and proteins are used as substrates for the energy-making process. They can skip glycolysis and enter the process at pyruvate, the overall product of glycolysis. So they skip glycolysis by entering in as pyruvate. Fats and proteins can enter in basically at step 2, the Krebs cycle, either as A-CoA, acetyl-CoA, or as pyruvate, to then feed into the Krebs and the electron transport chain where the majority of our ATP is produced in the mitochondria. Okay, thanks for listening and taking great notes during this discussion of Chapter 3. This was the last half of Chapter 3, and next time we'll discuss and diagram the functional aspects of the cell membrane in Chapter 4 and discuss the properties that you will experiment with in lab number 2, assigned for Monday and Tuesday, June 8th and 9th, depending on which lab you have. Thanks for listening.